Thanks to the organizers, Sergey, Elodie, and others for this wonderful conference. So I'm going to talk about at least I hope 2 topics, and if I make it in time, maybe even a 3rd topic. I will start with telling you about fold-seq, and as this also fits, I think, best into the framework of the conference. And this is a collaborative work. So actually, I discussed right after we learned about the results in CASP, 14, about the spectacular successes of AlphaFold 2. I zoomed with Marty and discussed what this means for us, and we decided we should develop a fast structure comparison algorithm, because there was nothing around that would be able to search these what we thought would be millions of structures. And so we basically started this project together, and I must say I have kept completely on the more theoretical side, whereas Martin has basically taken over completely all the practical stuff. So he's the one who does who handles all the bug requests and so on, together with my former postdoc Milot, who has done a lot, but he has moved now to Martin's lab, and he's doing a lot of the development and debugging and user support and so on. And then the first authors on the paper that hopefully will come out finally in Nature Biotechnology are Michael van Kempen from my group and Stephanie Kim from Martin's group. So structural alignments are interesting because often you don't have the sequence similarity that you need to a certain homology, and homology is of course what you want to have to do homology inference to predict function, structure, and so on. So in this case, this is ubiquitin and sumo. They are related by maybe 2 billion years of evolution or so. So there's 2 little tags of protein tags that tag other proteins. But their structures are actually very, very similar, despite these 2 billion years of evolution. And so the idea is that instead of using sequence searches, it would be much nicer to be able to do structural searches, particularly if we now basically have a structure for every or almost every protein of the uniprot. And so that was the goal. And as I told you, we were expecting there to be a big shortfall of available tools. And what you can see here is the time it would take. Actually, this seems, this is the right story. The time it would take with a 300 amino acid protein structure to search through the current redundancy filtered database of AlphaFold structures. So using the UniProt 50 and the high quality structures from Magnify ESM and PD1B100. So these are roughly 100 million structures. It would take DALI almost a year, TM-Align months, and fold-seq does this in 5 seconds. So that was not what we expected in the beginning, but this is how we started off. And the idea was so Martin and I had developed this MM6 software suite together. And it's a very fast and quite sensitive tool. So we were thinking, let's try to turn structural prediction into, or structural search, structural comparison into a sequence search problem. And of course, many people had that idea, and it's actually a 20, 30-year-old idea. One of the methods I've just shown here. So what all these people have done, really without exception, there are maybe 8 or 10 or so different types of methods that all rely on discretizing the backbone structure, so the C-alpha backbone structure of the proteins. And this is one called CLE. And you see that they have 4 different states for alpha helices, 4 for sheet-like structures or strand-like structures, and then the rest for coil-like structures, for loops and so on. The problem that we saw here, luckily, is that it's actually containing, using most of the information where there's least conservation, and using least of the information where there's most conservation. So most conservation is, of course, in conserved cores, where you usually have helix, 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 and least conservation is in the loops, where you have all these nice structures that are not really well conserved. So our idea was, in order to improve this, let's use, so maybe I should first finish this idea. So the idea is, of course, if you can write structures down into a sequence where every letter encodes a piece of the structure, then you can just do sequence searching. You can then take many structural alignments and do statistics and extract a substitution matrix like the Blossom 50 or PAM matrices, and then you can use standard BLAST or MM6 to do your structural searches in sequence space. So as said, this was in our, we had the feeling that this can be done much better, and of course, one thing that in hindsight I would say looks rather obvious is that what is much better conserved than backbone is tertiary interactions. So this is a very simple idea and it worked extremely well. So what we did is we said, let's take for every residue the nearest neighbor in 3D space. So in this case, this would be the blue one here, that is the nearest neighbor to the red one. And let's describe this interaction. So this, the 3D conformation of these little pieces of backbone. And so you can describe them with angles and distances. That gives you something like 10 dimensions or so. And then in principle, you can use k-means clustering and cluster these 10-dimensional vectors, and then you have an alphabet. And this we did, of course, but actually it turned out that it can be improved drastically in two ways. The first way is, instead of just taking the nearest neighbor by c beta distance, you basically optimize how the nearest neighbors are picked, because you want to pick nearest neighbors that are far in sequence. You don't want to have the situation that usually you pick I-1 or I+1, right? So you want to pick something that is 100 residues or so away. And for that, we just defined a virtual center that's basically roughly going in the direction of the C beta, but a bit further out and maybe a bit different. And using these virtual centers, we pick the nearest neighbors. Once we have the nearest neighbors, we can extract these geometric descriptors. And using these descriptors now, instead of doing k-means clustering, we trained a variational autoencoder. And so for the encoding of the sequence, you just need the encoding part. I will explain to you in a minute what this encoder does. So it basically just is a dense layer that ends in just 2 dimensions. So this is the Latin space, 2-dimensional Latin space. And then you have 20 vectors in this Latin space, and each vector stands for one of the 20 letters that we discretize into, and whichever is nearest will be picked. Right? So this is one of the variants of a discrete autoencoder. So how you train this is, there's also a trick involved here. So the standard to train a variational autoencoder is that you try to describe the vectors that you put in as well as possible by the output distribution. So you encode, you get these 2-dimensional Latin space vectors, you flip on or snap onto the nearest state, and then the nearest state, this is just 20 different points in this 2-dimensional space. This gives you, by the decoder, which is also just a 2-layer dense network, gives you parameters for the Gaussian distribution that is centered around the values that you think this input structure should have. But now instead of trying to predict the input structure again, which variational autoencoders do by default, we made use of the fact that we have homologous structures that we can align, and we want to learn particularly states that are as well conserved as possible. So we need to put in information about conservation. And this information is contained in pairs of homologous residues, where one belongs to one structure and the other to the other structure. So what we did here is that we used these pairs of aligned residues, so I and K. And one thing that I should point out is we don't only use geometric descriptors, we also use sequence distance. So these are just 2 monotonous transformations of the difference between the index I in the red one here and the index J, the blue one. And so running this variation autoencoder and using this alphabet actually improved the results quite a lot over simple k-means clustering. And I think the main reason is this conservation that is taken into account. So states that we learn are maximally conserved in a way. And so this is the 2D space that I showed you, the Latin space, and you see here the 20 vectors, and each one each dot here is a residue that was, or residue pair that was encoded, and you see two main axes here. One is the I-J sequence distance. So this is relatively trivial, but it turns out to be extremely informative. So and the reason why it's so informative is that if you have a multiple sequence alignment where one residue comes after the other, then in the homologous structures, almost always the aligned one comes after the other, right? So the order is not usually changed. So probably we are not very good in finding circular permutations, but that's a price that we can easily pay for the high information content we have here. And the other degree of freedom here in this two-dimensional representation is basically secondary structure type. So whether it's sheet or whether it's helix or coil. And in between there are different variants of sheets and coils, and also mixtures where one is a sheet and the other is a coil and so on. You can see here examples of these 20 states, and you see they're always coming in pairs. One is the I residue and the other one is the nearest neighbor interacting with it. And then we can learn a substitution matrix like in a standard amino acid alphabet. You see that it's very high information, has high values on the diagonal and usually very negative ones on the off diagonal. And using this alphabet, coming back to the example of ubiquitin and sumo, we see that now the sequence identity is not 16%, but it's 54% for the encoded sequences. So we call these 3D interacting sequences. So to make this fast, we used MM6, and MM6 you will now run just with these different sequences. It doesn't care what sequences it is. You give it the substitution matrix, and we had to tweak a few things. So, but most importantly, MM6 is a very fast algorithm because it has a very fast and very sensitive prefilter. So this part here is actually the main part, and it basically reduces the sequence space by at least a factor 1,000, and then it has a slower. So it looks at two consecutive similar k-mers. In Foldsig it's 6 mers that must occur on the same diagonal, and only those sequences, target sequences in the database that make it through this filter will get gaplessly aligned, and this gapless alignment is still very, very fast. Everything is done in AVX2, so very highly parallelized, and only when it's a certain score is overcome here, then you do a full Smith-Waterman alignment, and also that is of course AVX2 parallelized. And on byte level even. So this worked very well, and here you see the results as they're in the paper. So here we benchmark all kinds of different tools. So Geometric is a very fast tool for a bag of feature descriptors. This is not the standard precision recall curve, but it's similar. So what you see here on the x-axis is the fraction of queries for which we found this percentage above the first false positive. So for example, this dot here, this point here means that 85% of the queries found at least 75% of the, it's the other way around. 75% of the queries found at least 85% of the possible homologs. So in this case, of the same members of the same family. And you have, in scope, you have family level, superfamily and fourth level. So superfamily level is usually relatively difficult already to find. And so you see here in the comparison of these tools, the more on the right and upper side, the better. You see that Foldsig is more sensitive than a structural aligner called CE, a bit less sensitive than TM-Align and DALI. So there's a web server that is also quite highly used now, where so maybe I should switch back, where you can put in your PDB sequence, you press search after choosing a database, you get a result, you can click on each of the alignments, and you can look at the structural alignment in a viewer, you can highlight parts that will be highlighted in the structure, you can look at the aligned or non-aligned parts, and so on. You can also click to see this in a larger window. So this is work by Cameron Gilchrist from Martin's group and Milot Medita from Martin's and my group. Yeah, this is the structural viewer that is linked. And so now what I want to tell you about the newest developments is that we have, of course, tried to improve Foldsig further. And there was also a question yesterday whether you can do these searches not only with 3DI sequences, but also with 3DI profiles, and this is indeed the case. So MM6 has a nice advantage over Diamond, which is also a really nice and fast tool. that it can use sequence profiles for searching. And the reason is it does not need exact matches. It never uses exact matches. So for example, these k-mers, these two k-mer matches here, they don't need to be exact, they can be similar. So what we do is for every k-mer in the query, we generate a list of similar k-mers. And these lists of similar k-mers can be either generated using the Blossom matrix, in which case we have a certain score cutoff up to which we generate these similar k-mers, or we can generate them from a sequence profile. And it's actually basically almost the same thing. And it means that this the pre-filter works basically the same for profiles as for sequences, and also these alignment algorithms, they just use profile scores instead of Blossom or substitution matrix scores. So everything is basically the same, and that means that we can now run profile searches with 3DI profiles, and this is the plot that I just showed you. And if we now look at what happens if we do one iteration through the structural database, you see that we are already getting almost as good as TM-align, and then with 3 and 4 iterations, basically we're at the same level of sensitivity as TM-align, and only very slightly below DALI. Okay, so the next steps. So maybe I should point out that these alignments are, I think, very useful for homology detection and annotation, because they don't care about steric conformational conformations of domains with respect to each other. So often you have the problem that TM-align and DALI and so on, they cannot find superpositions. DALI is a bit better, but TM-align has a problem finding superpositions that include two domains that have a different conformation. And of course, FoldSeek doesn't care, because it basically only looks at the internal logic of each of these folds. The applications, I think, are very, very numerous. Basically, a lot of things that you, or maybe most of the things that you could do with amino acid sequences, you can do with 3DI sequences. For example, many people write to us who want to do phylogeny or functional annotation and so on. And the nice thing is now that this is also only slightly slower than amino acid based searches. Yeah, and the next steps for us are, we want to have a better, even better alphabet, and we have a collaboration ongoing with Jeanne Trinquet, who is in the audience and who will give a talk later, and Sergei Ovchinnikov, with whom she did an internship. So this is, I think, a very elegant method that you'll hear about, and very different from the one that we used here. And we're also working on developing a better structure similarity score that will help us rank or re-rank the structures. So basically, we want to use not only the information from the nearest neighborhood, we want to use the whole structure, the whole structural alignment to get a better ranking. And then there are, of course, other things that we are working on. Yeah, and if I have enough, how much time do I have?
2 minutes.
2 minutes. Then I will talk about Roshi's project, and then I'll stop. So one way we apply this is with in a tool that's called SpaceStas. Here the goal is very different. So in principle, this doesn't have anything to do with protein structures, but we use FoldSeek to enhance it, basically. So the idea here is that a lot of information on the function of proteins for prokaryotes, for viruses, and for some simpler eukaryotes is contained in their genomic context. So often proteins that work together with each other in some functional module have a conserved or partially conserved gene neighborhood. And you can see an example here of an antiphage system that consists of 6 or so genes. And you see that this is a result of the search of Roshi's tool that gives you similar clusters. And similar means it can be very widely varying. The more proteins you have, the more sensitive it will be to find things that even contain only a fraction of those. If you have only 2, then basically it finds only those that have the 2, maybe with a gap or maybe with a strand switch. But the more proteins you have, the more similar clusters it will be able to find. And the interest that we have in this is, of course, that we want to use it for annotation, or to help people with annotation, and what I would call horizontal annotation transfer in contrast to vertical. So vertical meaning homology, like in an alignment where you vertically list the sequences. And here you basically go from one annotated or maybe 2 annotated proteins in a functional model, and you guess what the other proteins could be. And one example is here, I think this is inulin. Yeah, inulin is a polysaccharide utilization model where you see there's a lot of stuff in, and that is closely linked to this pathway. So this is a relatively old idea, but funnily there is no real fast and flexible tool available to do these kinds of cluster searches, so similarity of gene clusters. So this is what Rochi's tool does, and the way it works is, so imagine you have here all the genes of a query genome, and here you have all the genes of a target genome, and you've done an MMSeq search where you found all the homologous pairs of one genome with the other. And let's say you match this gene here from the query with this gene in the target, and this with this, and so on. Then basically what the algorithm does is it looks for clusters of genes that are maximally significant. And we measure significant according to 3 criteria: how strongly they are clustered, so how small this rectangle is in which we find them, how conserved the gene order is, so whether there's something in between, or whether it's 1, 2, 3 versus 2, 1, 3 or so, and how well the strandedness is conserved. And all these are described by p-values. And then basically it's a greedy algorithm that finds all these clusters with maximum p-value. And so when you can use this tool now to compare, if in a set of representative genomes, every genome with every other genome. And here we do the comparison not only with MMSeqs, but we are fetching structures from the UniProt. So we are searching with fold-seq, or we first searching with MMSeqs through the UniProt database, get the fold-seq structures, and then search with fold-seq the structural similarities in these genomes. So instead of using MMSeqs matches, we basically use fold-seq matches. And then we find these clusters. So here, this is from a query genome, the number of cluster matches that were found for a certain protein or gene with, for example, index 100. So that would be 75. This would be part of 75 cluster matches. And then we can take a cutoff, and I think I have to finish, right? and look at what we found. And to finish this off, I don't have time anymore to talk much about it, but you find a lot of interesting conserved clusters. This is just one example. Is a light harvesting system in cyanobacteria, and you see they're very nicely clustered and have a conserved, more or less conserved structure. You find all these antiviral defense systems. Some of them only partially. And some of them not. These are all the ones that have only 2 members, but the ones with 3 members basically can all be found. So, and this is de novo. So this is actually what we compared to is a system that is only specialized on these antiviral defense systems, and we can find them de novo. And we of course find many other things that have maybe never been looked before. So we are now in the process of analyzing this data set. And there's a Colab notebook available, and you can already use this, and I give a link on the last slide. So I'll just skip over Annika's project, a new assembler for viruses, but this anyway doesn't fit so much into this topic. And I thank everybody for your attention.
Thanks a lot.
Thanks a lot, Johannes. Technical question, why 20 letters and why 2-dimensional representation, not 3D, not 4D?
Yeah, I think it doesn't make so whether 2D or 3D doesn't make a big difference, because it's just, there are just 20 states, and you have 2 hidden layers, so you have enough flexibility, I think. Probably, I mean, I'm not sure whether we've tried 3 or 4 dimensions, but we've tried, of course, other numbers of letters. And there it's just a sweet spot for our MMSeq algorithm. So you have to generate these similar k-mers, 6-mers. And if you have a larger alphabet, let's say 32 instead of 20, you have to generate a lot more, because basically you are losing a factor 6 to the power of 1.5 or so for every 20 to the well, you're losing a big factor exponentially, basically. So if you go to lower numbers, then you're losing too much sensitivity. But with 20, the lengths of lists that we generate of similar ones are still not dominating the runtime, and therefore it's basically a sweet spot.
So I have a question.
if you could elaborate a tiny bit on are there any cases where you find things with the fold-seek algorithm that are that according to fold-seek are similar, and in reality perhaps they're not all that similar, and the reverse where you miss similarity with this approach?
That's a good question.
And is my intuition correct that any gaps, I mean structurally gaps are probably more difficult to justify than with sequence alignments, that they're actually really in the loops, right? I mean, that's the only place that they could be. Okay.
Yeah, so I think that the limitations are, particularly with respect to the structural aligners, are really very highly diverged structures. So ones where maybe the nearest neighbor is not conserved anymore. So you pick a nearest neighbor, but in the homologous structure, it's actually in a different one. Then, of course, these two states will be very different. I think this is certainly a limitation. But my guess is that this is what we are improving on using the profiles.
But wouldn't the next one catch that? I mean, you're not just relying on
so far, we are only relying on the next neighbor. So this is, you could call it a weakness. But I think this is also why we're interested in getting a better 3D-I alphabet that would not have this weakness. So what Jeanne will tell you about is how she describes these neighborhoods using a neural network that basically through message passing summarizes a lot of information about the environment. And it's end-to-end learning and very elegant, and this, I think, addresses this question. There are actually quite a few cases where fold-seek works better than TM-Align and DALI. So basically compensating the cases where it's not as sensitive. And those are, for example, cases where you have rearrangements or warps, where the structure is flexible and particularly TM-Align does not cope very well with that. And there's also, so what I didn't show is DALI does not look very good in the precision recall plot, and that's because it does not calculate good significance values. So the Z values that you get in DALI actually are not really good to compare one search against the other search. So that's also where AlphaFold is actually really good. So the significance values, so the probability and E values are very very reliable and comparable between different searches and different structures.
Beautiful talk and great work. I had a question. Clearly for the search application, it makes sense to throw away the side chains. But if you're doing some kind of evolutionary phylogenetic or some of these other applications, wouldn't you want to also discretize the backbone rotamer library and somehow incorporate that into your 3D representation of structure? Could you still get a sequential representation by doing this?
Yeah. I guess that would probably make sense. I mean, for the moment, we have focused on being as fast as possible. So we could not really put more information in there than 20 states can describe. But for other applications, for example, if you want to do phylogenetics, it might make sense to learn an alphabet of size 1024 or something. And then it might make sense to encode more information in these states.
Do you see any barriers other than speed to doing this?
So if you use 1024, it will be very slow with MM6, but for calculating a phylogeny, that might be still fast enough. So we haven't thought in detail about this yet.
Okay, thank you. Let's thank Johannes again. And